Hello, everyone. I'm Tim McCay from Colgate University, and today I'm going to talk about one part of the jumping worm invasion story, what these worms may be doing to the other animals that live alongside them at the forest floor. The original research that I'll be talking about today was done with my co-authors, Sophie Pedzich and Edward Cherry, and that work focuses in central New York and the Northeastern United States. So when people first encounter jumping worms, the worms themselves naturally get most of the attention. They can be really abundant, they move vigorously, and at heavily invaded sites, they can noticeably change the appearance of the forest floor if they're in a forest. But I'm most interested in what is living around them. The forest floor supports a remarkably diverse community of animals. So if jumping worms are changing the leaf litter habitat, we need to ask whether they're also changing the biodiversity that depends on that habitat. Going into this study, I expected that they probably were, but as you'll see, the answer that we found was not quite that simple. So I'll begin with a quick overview of forest floor biodiversity. Then I'll talk about earthworms as ecosystem engineers and what we already know about the effects of invasive European earthworms. And after that, I'll turn specifically to jumping worms and in particular, what we did, what we found, and how I think we should interpret the results. So when we look down at the layer of dead leaves in a forest, it's easy to think of that material as just debris. Ecologically, though, it's a very active habitat. There are mites and springtails, spiders, pseudoscorpions, centipedes, gastropods, nematodes, fungi, and many other organisms. And they're all doing a great deal of ecological work. They contribute to decomposition and nutrient cycling. They disperse fungi. Some are even involved in pollination. Some are herbivores, some are predators, and many are prey. Salamanders, birds, small mammals, and large invertebrates all feed on these animals that live in the litter. So the forest floor community connects the decomposer system at the bottom of the food web with larger animals above it. The litter also matters as physical habitat. It holds moisture, it buffers temperature, and it creates a complex three-dimensional environment. So when something changes the litter layer, it has the potential to affect much more than just decomposition. Earthworms are really effective at changing that layer. Darwin recognized this long before ecologists even started using the term ecosystem engineer. This photo comparison shows the effect pretty well. On the left, where earthworms are absent, there's a thick organic layer, including multiple years of incrementally decomposed material. And on the right, where earthworms are abundant, there's a much thinner litter layer consisting almost entirely of leaves from within the last 12 months. As you may know, many northern North American forests developed after glaciation without large litter processing earthworms. So when invasive worms arrive, they introduce a process that had been absent since the origin of these ecosystems post-glaciation. And that means they can have unusually strong effects on the structure and the function of forests by affecting that soil layer. Jumping worms are part of this larger earthworm invasion story, but they're not, as you may know, just one species. When biologists say jumping worms, at least in the Northeast, they're really referring to three species: Amynthus tokyoensis, Amynthus agrestis, and Metaphyra hilgendorfi. These species are similar, but pretty consistently different in size. They're called jumping worms because of the thrashing way that they can move when they're disturbed, but their ecological importance comes mainly from where and how they feed. They live close to the soil surface, and they can occur at high densities. At heavily invaded sites, the soil surface may develop the loose granular casting material that people often describe as resembling coffee grounds or cooked ground beef. Another aspect of their biology that's important is that they are annuals. They overwinter as eggs, they hatch in the spring, and they grow through the growing season. So their effect might be expected to vary depending on the time of the year. And I'll say more about that later. So these worms are actively modifying the same habitat that's used by the litter animal community that I just talked about a moment ago. And that gives us a pretty straightforward research question then. What happens to those other animals when jumping worms enter the system? There's already a substantial literature actually on invasive European earthworms in northern forests. So one way to frame the jumping worm question is to ask, what would we predict based on those earlier invasions? So let's look at what studies into European earthworm effects have found. This is one example from work that Peter Scull and I published in 2019. The horizontal axis shows earthworm biomass, European earthworm biomass, and the vertical axis shows the total abundance of litter animals. And the relationship between these things is pretty striking. Sites with little or no earthworm biomass include very high densities of litter animals, but at sites with high or even moderate invasive earthworm biomass, those high litter animal densities disappear. So in this study, greater invasive European earthworm biomass was associated with far fewer animals in the leaf litter. And we found that the actual mass or volume of that litter was the probable intermediate factor. These earthworms reduced the litter amount, and in turn, litter animals had less habitat and they declined. And that study is not an isolated example. A number of studies have reported substantial declines in forest floor animals associated with European earthworm invasion. The exact response differs among studies, but some of the effects are large, 75%, 70%, 60% decline in abundance, for example. In particular, microarthropods, small arthropods, including primarily mites and springtails, have declined sharply in some invaded forests. So when European earthworms invade forests that historically had few or no earthworms, the forest floor animal community often declines. That's well established. Earthworms remove litter habitat, they change moisture conditions, and they change the distribution of nutrients. They also can affect fungal and bacterial distribution. So sometimes the mechanisms of that earthworm effect can be complicated, but the general association is clear. And so this gave us a pretty straightforward research question and hypothesis to work from with respect to jumping worms. Like with European earthworms, we would expect that jumping worms might negatively affect the abundance of litter animals through changes in the forest floor habitat. And for jumping worms specifically, the effects on the litter animal community have not really been studied directly in much detail at all. In some sources, the effects of European earthworms have been presumed to be the same as those for jumping worms. But we cannot really presume that, especially given some of the differences in biology between these groups that I mentioned a moment ago and that I'll highlight again here in a little bit. So the basic question that we asked in the research study that I'm going to tell you about is, do forests with jumping worms have fewer or different forest floor animals than forests without jumping worms? Our approach to answering this question was a comparative field study. We selected sites with jumping worms and sites with few or no earthworms and no jumping worms. And at each site, we sampled the earthworm community, we sampled the litter invertebrate community, and we measured the amount of leaf litter. And for part of the study, we also measured environmental variables, especially soil pH and soil moisture. And those environmental measurements will become important, as you'll see. This was an observational study, obviously, so we correlated all of these things to better understand associations and to start to make some first stabs at cause and effect. These are our study sites around Hamilton in central New York. The red points are sites where we found jumping worms, and the tan points are sites where we did not find them. Because these sites are all in the same general region, they were all broadly similar with respect to climate and forest type, but they're still real forests with different histories and different local conditions. In particular, the geology varied a bit, and this might have affected the earthworm community, as you'll see. At each site, we sampled 4 quadrats separated by 5 meters. The yellow square represents the larger earthworm sample. The smaller dark square inside it represents the litter and invertebrate sample. Using several sampling quadrats like this gave us a better picture of conditions at each site than a single quadrat would have, but the site in its entirety, including all of these quadrats, remained our true biological replicate for purposes of analysis. For the litter fauna, we collected a standardized 25 by 25 centimeter sample of leaf litter. We brought those samples back to the lab and placed them in Berlese funnels, which are used to extract animals from those samples. These funnels dry the leaf litter from the top, and as it dries, animals move downward, and eventually they fall into collection containers. We then sorted the material and identified animals generally to order or subclass. For the earthworms, we sampled a 60 by 60 centimeter area, and we used hand collection as well as something called mustard vermifuge, which is using a spicy mustard solution to irritate earthworms in the soil and bring them to the surface where they can be collected. We identified all the worms that we collected to species and also estimated their dry biomass from their body lengths. So the data that I'm going to show you were collected during 3 sample periods. The first samples were collected in the summer of 2022. And for those sites, we measured the worms, litter, and invertebrates, but we did not measure any environmental covariates, anything beyond the community. We then sampled additional sites in autumn of 2022 and then different sites in autumn of 2025. And for those autumn samples, we also measured soil pH and moisture. So this created 2 related data sets. There's a full data set, including both the summer and the autumn samples, that gave us the largest number of sites for comparisons between jumping worms and non-jumping worm sites. But the smaller autumn data set additionally allowed us to include pH and moisture as covariates in the analysis. So this distinction is important because the apparent effect of jumping worms might change if other factors are also considered in this kind of a correlative study. So I'm going to present the results of this work in 4 stages. First, I'll show you what we actually found in the earthworm samples and whether the site selection approach worked. Then we'll look at the amount or mass of leaf litter and how that might have been related to jumping worm presence. Third, we'll focus on those environmental covariates, particularly pH, to see what relationships those things might have had with jumping worms. And finally, we'll turn to the invertebrate data to see if the invertebrate community was different in places with and without jumping worms. So we'll first use the full data set without covariates, and then after that, we'll turn our attention to the smaller autumn data set where we can account for pH and moisture. So the first result is that our site selection approach worked pretty well. We had 10 sites where we only detected jumping worms, no European worms. We had 10 sites where we detected no earthworms at all. And we had 2 sites where we found European earthworms, but no jumping worms. The difference in earthworm biomass was also pretty striking. The jumping worm sites' average dry biomass was close to 4 grams per sample. At the 2 European earthworm sites, the biomass was only about 1/10 of a gram. So the jumping worm sites had markedly more earthworm mass at them, and also gave us some indication that they were supporting meaningfully large jumping worm populations. So given this variability in jumping worm abundances, our first ecological prediction was probably the most obvious one, and that is if jumping worms are consuming surface leaf litter, then invaded sites should have less leaf litter. And the sample average did go in that direction. Jumping worm inhabited sites averaged about 176 grams of litter compared with about 200 grams at sites without jumping worms. But there was a lot of variation, especially among the sites with jumping worms here on the right. And statistically, the difference wasn't convincing. So, based on this comparison, we could not conclude that jumping worm inhabited sites had substantially less leaf litter. And that result surprised us for sure. But to better understand this, we then focused our attention, okay, just on the 10 sites where we had jumping worms to see whether the abundance of jumping worms might have meaningfully explained the amount of leaf litter at the sites. And we did find generally as there was more jumping worm biomass that we found less leaf litter. But probably the most important thing to observe here is that there's a strong difference between the sites sampled in the summer, the red dots on the left, and the sites sampled in the autumn, the blue dots on the right. Remember that jumping worms are annuals. So in the summer, populations and individuals are small and maybe not capable of much of an effect. Sites sampled in the autumn were all clustered here around 100 grams of leaf litter. So just to go back to this side, those are these points here at the bottom of that distribution here, pretty convincingly different than the distribution of sites without jumping worms. So jumping worms in the autumn seem to be having an effect, but when we sample jumping worm inhabited sites in the summer, it gets a little bit messy. So anyway, at this point, this is a useful thing to keep in mind regarding the seasonal variation in these earthworms and how it might be affecting leaf litter dynamics. All right, turning our attention to the environmental correlates now, pH and moisture. We found that jumping worm biomass was strongly related to soil pH. We never found jumping worms at sites much below pH of about 6 at our sites. And the higher the pH, the more jumping worm biomass we tended to find. Moisture, in contrast, was not convincingly related to jumping worm biomass, at least within these sites. This pH effect is important because other litter animals also respond to soil pH. And this creates a potentially confounding problem. So if we find an association between some type of animal and jumping worms, we really need to tease out the potential role of pH in affecting both jumping worms and that other kind of animal, if we want to attribute the effect directly to the action of jumping worms. And so with that, now we'll turn our attention to looking at the invertebrate groups. So I have these listed. And I'm starting here with a raw comparison using the full data set, including both summer and autumn samples. And the first thing to notice is that among these first 5 groups, that there's no broad, convincing decline in abundance of these animals at sites with jumping worms. In fact, several of the groups have higher mean sampling abundances at the jumping worm sites. Even statistically significantly higher means for gastropods, which includes the slugs and snails, and mites. The NS here for isopods, spiders, and pseudoscorpions indicates a non-significant difference. But you can see that the sample means are differing in both directions, higher and lower with jumping worms. So I'll return back to this gastropod and mite association, positive association with jumping worms in a second. So there are more groups here: centipedes, millipedes, springtails, beetles, flies, true bugs, ants. None of these are convincingly different statistically. And the sample means, as you can see, are differing in both directions. Sometimes the sample means are higher when at the sites with jumping worms, and sometimes they're lower. And then this is the last of the three slides that encompasses all the groups that we looked at. Caterpillars, bark lice, and thrips, all of these groups had higher sample means at sites without jumping worms in the direction that we might expect, but not convincingly so statistically. So just based on that broad analysis, including both the summer and the autumn sites, we found no broad decrease as expected with jumping worms. But we found a couple groups, gastropods and mites, that actually were convincingly more common with jumping worms. And if indeed jumping worms were making the world better for even a few groups, that would be surprising for sure. So we wanted to dig into that. Before we can conclude that, though, we need to consider soil conditions, which we saw, at least for pH, can co-vary with jumping worms. So now we're going to look at the same sorts of comparisons, but now just for the autumn sites where we also had pH and moisture data. So our analysis here allowed us to parse out the effect of soil conditions, pH and moisture, from the effect of jumping worms. So here we see that although there are indeed more gastropods and mites, where we also find jumping worms, that difference is most strongly due to soil pH, not to the jumping worms themselves. So what we're really left with regarding these groups then is a lack of compelling evidence of an effect of jumping worms on these groups, really one way or the other. We've got pH and perhaps moisture for mites affecting the abundance of these groups. But you'll notice the NS under that jumping worm effect column is really indicating that jumping worms were not a significant predictor of the abundance of these groups one way or the other. Right, just like before, we've got a few slides to complete the full lineup. For most of these groups, centipedes, millipedes, springtails, flies, we again find no clear jumping worm effect. Beetles, interestingly, are the one group here where the model does suggest a possible jumping worm effect. They were somewhat less abundant at jumping worm invaded sites, and the test falls right around the conventional 5% significance threshold. I want to be a little cautious about interpreting that result. It's marginal. We did a lot of tests here, but it does provide a useful hypothesis for future work. Particular beetle groups or beetles generally may be sensitive to jumping worm invasion, even if the broader community doesn't show a strong response. And for these final remaining taxa, there's again no convincing jumping worm effect, but again, pH continues to appear important for some of the groups, caterpillars and maybe hymenopterans, which includes mostly ants. So before we leave pH behind, I want to say that the importance of soil pH in this story really stimulates some additional logical questions. Are forests with more basic soils more susceptible to jumping worm invasion? It seems like that might be true from this analysis. Are the worms themselves contributing to changes in soil chemistry? That's been demonstrated for some other earthworm species. And are both of these things happening, both a push and a pull effect of the worms and their relationship with pH? These are interesting hypotheses for future work, but for now, we at least have to conclude that future studies of jumping worms should always consider the importance of pH, soil pH, in both how they're responding to and how they're affecting their environment. So far, we've considered one taxonomic group at a time, but it's possible that jumping worms might change the overall community, even if no single group shows a dramatic response. And this is what this ordination examines. The details of this NMDS method are less important here than the basic interpretation. Each of these points is a site, and sites with more similar invertebrate communities appear closer together in this two-dimensional plot. The red points are jumping worm inhabited sites, and the open points are sites without jumping worms. And if jumping worms caused a consistent shift in community composition, we would expect these two groups to separate clearly. But the separation is not super convincing, especially given the variability that we found in jumping worm inhabited sites and the two sites that are depicted on the right-hand side of this graph. There's a lot of overlap. So at least at this broad taxonomic resolution, we're not seeing a clear community signature of jumping worm invasion. All right, so let's get to some interpretation. Why did we not see the strong effects of jumping worm activity on leaf litter animals that we expected? Well, one possible explanation is in the annual life cycle of these worms. Juveniles appear during the spring, they grow through the summer, adults are abundant by late summer and early autumn, then the adults senesce and die, and the population overwinters as eggs in little cocoons. Now think about what happens in the forest at almost the same time that the adults are disappearing. The leaves fall. So just as the active adult population of jumping worms is declining, the forest receives an entirely new layer of leaf litter. And that litter then remains through the winter into the early spring when jumping worms are absent or very small. So the forest floor may be depleted during part of the growing season and then rebuilt in the autumn. This annual life cycle may therefore limit how completely jumping worms can remove the litter layer over the long term, and that could matter for invertebrates. The conditions experienced by the litter fauna in August may be very different from the conditions in November or April. So a one-time comparison among forests may miss an important seasonal effect. We had some samples in the summer, some in the autumn, but this study admittedly lacked the seasonal resolution to really understand this seasonality. So what can we take away from this study? First, we didn't find strong evidence that jumping worm sites had substantially less litter, but this was complicated by that seasonal variability, as I said. Secondly, we didn't find a broad decline in the abundance of invertebrates that depend on that leaf litter. Most taxa showed substantial overlap between invaded and uninvaded sites. Some groups were actually more abundant at sites that were inhabited by jumping worms, but those differences were probably due primarily to soil pH. And third, we didn't find a clear difference in the overall community composition of invertebrates. So the simplest version of the hypothesis that jumping worms arrive, remove the litter, and cause a general collapse of the litter animal community was not supported by these data. But I also don't think that the correct conclusion is that jumping worms have no effect. There were several important limitations of this work. First, we did not know how long jumping worms had been present at each site, and the length of invasion may have been important. Second, as I argued here, and perhaps of primary importance, is that the effect of jumping worms might vary a lot through the year as the worms move through their annual life cycle. And finally, our taxonomic classification was very coarse. If one fly family declined while another increased, we might have seen little change when all flies were combined in our analysis. So I really think that the next step needs to be a finer scale examination of this association. We need repeated sampling throughout the year at sites with good information on invasion history, and we need a finer taxonomic resolution of litter animals and how they might be associated with jumping worms. So the message is not that jumping worms do not matter. It's just that their effects on litter biodiversity may be more subtle, more seasonal, and more dependent on local forest conditions, particularly soil pH, than we initially expected. So I'll finish by thanking the people that helped to make this work possible. The project was supported by the Colgate University Department of Biology. A number of students helped in the field and laboratory. And I also want to thank the Healthy Soil Collaborative for bringing together researchers working on different aspects of the jumping worm invasion. And finally, I want to acknowledge my co-authors, Sophie Pedzich and Edward Cherry, for their contributions to this project. Thank you.